Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video, we're going to be continuing our histology lecture series talking about immunohistochemistry and the histopathology laboratory. All right, let's get started. Immunohistochemistry is an area in the histology laboratory that is very important for assisting pathology and making a diagnosis. It is used to determine the origin, prognosis, and treatment of a patient's tumor. Immunohistochemical techniques require rigorous quality control, and this ensures that the results produced are accurate and consistent with staining patterns that are expected. A major role of the immunohistochemist is to troubleshoot variables that can occur due to fixation, processing, and various tissue types. It's of course important to understand the basics of immunology, so we'll be discussing that in the coming slides here. So immunology involves antigens and antibodies. Antigens are molecules such as proteins, polysaccharides, other polymers, and nucleic acids. These substances can induce a detectable immune response when introduced to, in our case, in histology, a human. So the most common antigens we encounter as humans are bacteria and viruses. So for example, a virus antigen invades the human body. It's a foreign antigen, meaning that the body doesn't recognize it. And so because it doesn't recognize it, it sees it as a harmful invader. And so the human body then launches an immune response to it. And part of that is by the B lymphocyte white blood cells producing antibodies that are specifically directed towards that foreign antigen. Antibodies are host proteins, also called immunoglobulins, and there are 5 major classes of them. And we call those IgG, IgA, IgM, IgD, and IgE. So again, antibodies or immunoglobulins are proteins that are produced in response to the invasion of a foreign antigen. Antibodies are Y-shaped in structure, as you can see in this drawing on the bottom right-hand side of this slide. So there are heavy and light chains on each antibody. And I stated on the previous slide that there are different classes of antibodies, and these classes differ in both structure and function. So IgG, IgA, IgM, IgD, and IgE. So there are 2 identical heavy chains, and that determines the subclass, so G, A, M, D, or E. The light chain is either kappa or lambda. Now you will see in this picture I created here, there's a constant region and a variable region. So the variable region is this yellowish color on the upper arms of the Y, and this is where the antibody binds to the target antigen. And this region of the antigen is called the epitope. So the region on the antigen that is recognized by the antibody here is the epitope. Now the pink area, or the constant region of the antibody, remains constant across different antibodies of the same type. So we use polyclonal antisera and monoclonal antibodies in the laboratory all the time, and also in histology. So polyclonal antisera are antibodies that come from animal blood that has been immunized with peptides or proteins. So that's a lot of scientific wording here. So what does this even mean? So what happens is an antibody, so specifically class IgM, is taken from a human, right? So the blood is taken out and the IgM is taken. So that antibody is then injected into a rabbit. So the rabbit's immune system sees that IgM human antibody as foreign. So meaning it isn't from that rabbit, it's a foreign substance. So the rabbit's immune system launches an attack in the form of its own antibodies. Specifically, in this case, it produces an anti-human IgM antibody, and it produces that anti-human IgM antibody because we injected the rabbit with human IgM, right? And so it recognized that human IgM as a foreign substance, so it launched an antibody directed towards that human IgM. So once it launches the attack with the anti-human IgM antibodies, the rabbit serum can be taken and used as a polyclonal anti-human IgM antibody reagent. So lymphocytes are a type of white blood cell, and in response to a foreign substance, each lymphocyte produces a cell line. All cells in this line are a single clone that produces an identical antibody. But various clones produce antibodies of different classes with the specificities to different epitopes on the antigen. So this results in a mixture of antibodies produced by many lymphocyte clones. This pool of antibodies is called polyclonal antiserum. And again, polyclonal antisera is used frequently in the laboratory. It is highly sensitive because of its ability to bind to many epitopes. Although it is highly sensitive, it's not as selective, which may result in some nonspecific staining. Rabbits are commonly used for the creation of polyclonal antisera, like this little cute one on this slide here, but also goats, sheep, horses, and humans can be used for the creation of polyclonal antisera. Monoclonal antibodies are created by injecting mice with an antigen. B lymphocytes, which again are a type of white blood cell, in this particular case, the B lymphocytes are fused with non-secreting myeloma cells. And myeloma cells are cancerous tumor cells that can divide indefinitely. This fusion causes the production of something called a hybridoma. The hybridoma retains the capability of antibody secretion, of course, of the B lymphocyte, and the immortality of the myeloma tumor cells. So we're merging and fusing together a B lymphocyte, okay, that can produce antibodies and secrete antibodies with a myeloma tumor cell that is producing, dividing things indefinitely. So once these hybridoma cells are created, they can be cloned. A single clone is capable of producing antibodies that are identical in molecular structure to the original. The antibody is able to be produced in unlimited quantities. So the main advantages of monoclonal antibodies are that they are something we call highly homogeneous, meaning they are the same. They are highly specific and of course can be made in very large quantities. Rabbits can be used for monoclonal antibody production, but the ones generated using mice are most common. Now that we've discussed what antigens and antibodies are, let's talk about how we handle the fixation and processing of tissue that is going to be undergoing an immunological procedure. Before any immunologic procedure, the stability of the antigen must be considered. Most antigens are soluble in aqueous solutions, meaning they dissolve in water. Because of this, the antigens must be fixed in situ, which of course the specific technique permits it. So this means it must be fixed in its original place. We'll talk about immunofluorescence in the coming slides, but this is an immunohistochemical technique, and in this technique, antigen reactivity must be preserved as much as possible. So this is an example of why antigen stability must be considered. So here's the example here. So for this technique, frozen sections of unfixed tissue are the classic preparation of tissue. So in this, the activity of the antigen is the least impaired, and fluorescent antibody staining is the strongest. Soluble antigens, if present, are lost with this technique. Tissues that have been fixed in formalin are rarely used in the immunofluorescent technique. This is because it provides inconsistent results because fixation and processing impair the activity of the antigen. So we're wanting to look at the activity of the antigen. So we want to make sure that we're using techniques that don't inhibit that antigen or prevent that antigen from being active. Dehydration of tissue and chemical fixation are two of the major categories of tissue fixation. In dehydration, the proteins unfold and their solubility changes. The fixation of tissues with chemical reagents denatures and stabilizes proteins by either coagulation or by forming additive compounds, or of course a combination of both of these. Acetone and alcohol are coagulating non-additive reagents. Formaldehyde is a non-coagulating additive fixative that cross-links proteins, and mercuric chloride is both a coagulating and additive fixative. The coagulant fixatives that don't form additive compounds allow good penetration of the antibody, and they do not block immunoreactive determinants. However, the treatment of cryostat sections with acetone doesn't achieve complete fixation. Extended immunohistochemical procedures produce morphological changes like chromatolysis and the loss of membranes. Chromatolysis is when a damaged neuron disperses the Nissl substance. For frozen sections that are going to undergo routine immunohistochemical staining, they should be cut from 4 to 5 microns and then placed in 10% neutral buffered formalin for 5 minutes. After that 5-minute period, it should be transferred to phosphate buffered saline. Now, if doing muscle tissue sections, they should be cut and allowed to air dried for an hour, then immediately be stained with antibodies targeted for dystrophin or other muscular dystrophy-related proteins. And as a note, glutaraldehyde preserves the morphology the best, but it irreversibly blocks antigenic tissue determinants, so it's not ideal for immunohistochemical studies. Now, if the tissue has been processed in paraffin, proper tissue handling is incredibly important in producing consistent results with immunohistochemical staining. Fixatives can positively or negatively influence the staining results with a given antibody. Not all antibodies are affected in the same way. Key factors that influence staining quality are less than 12 hours of fixation delay, so less than 12 hours delay in fixation, fixative properties of concentration, the pH and buffered solution, time in the fixative, and storage of mounted slides. The histotech must document the time the tissue was placed in the fixative. An example of why this is important to do is for breast tissue for HER2 testing, which is a breast cancer testing. So the tissue must be fixed at room temperature, a minimum of 6 hours with a maximum of 48 hours. So we need to make sure that we document what time it is in there, because we need to make sure that for this specific HER2 testing on this breast tissue that it has made the minimum but is not over the maximum. So that's why everything needs to be labeled with, documented with when it was placed in the fixative. If a laboratory is using multiple different types of fixatives, each antibody protocol should be validated with each of those types of fixatives to ensure that it works properly with them. Fixation in neutral buffered formalin causes hydrogen bonds to form. These must be reversed to ensure that the antibody can bind to the antigen site. In rare cases, the epitope cannot be retrieved, and in these cases, frozen sections will be required to be used. The best practice is for the histology laboratory to standardize the fixation time for specimens using the mildest retrieval methods that cause minimal damage to the morphology of the tissue. Zinc formalin fixatives preserve the immunoreactivity of the tissue well. It can be used on automated tissue processors, which is great. Many labs have switched from the 10% neutral buffered formalin to a formulation of zinc formalin for routine processing because of this. Mercuric chloride and Buin solution are rarely used, and the B5 is noted for producing excellent nuclear detail in bone marrow and lymph nodes. Because of mercury being an environmental hazard, it is being replaced with acetic zinc formalin, and this yields results comparable to the B5 in immunohistochemical staining. Buin fixative, which is composed of glacial acetic acid, formalin, and picric acid, is not ideal for immunohistochemical staining. Picric acid is difficult to remove, and crisp staining detail is lost for these stains. Fixation varies between laboratory to laboratory. Vimentin is a structural protein that makes up the cytoskeleton. Staining of vimentin provides a great way of determining when a tissue section has been overfixed. The staining quality of vimentin in paraffin sections is excellent, and the quality of the stain in paraffin sections progressively worsens as the length of time in the fixative increases. So when the vimentin stain is negative, the tissue is overfixed, and all antibody reactions should be interpreted with caution. If the vimentin staining is uneven, the immunostain should be read in the area of the most intense vimentin staining. So this is a good thing to use to ensure that the time and fixation has been appropriate. Complete fixation before the specimen is processed is needed in order for immunohistochemical stains to work optimally. Thorough dehydration and infiltration are needed for the stain to be consistent throughout the tissue specimen. False negative or variable staining in the middle of the tissue section with proper staining on the edges is considered an artifact. This is a common occurrence that is produced when the outer edge is fixed in formalin and the center is alcohol fixed, resulting in underfixation. Antigens are altered by high temperatures, and tissues that are exposed to a high temperature can be damaged. Reprocessing might also alter the epitope sites, which leads to false negative results. So this photo shows a lymph node tissue that is fixed with formalin. It's stained with leukocyte common antigen, which demonstrates an alcohol-fixed center. This results from an incomplete formalin fixation before beginning the processing steps. Proper microtomy and immunohistochemical staining tissue sections is important as well. The quality of the stain is greatly affected by the quality of the tissue section. Tissue sections should be free from wrinkles and folds. The water bath should be filled with clean DI water for immunohistochemical slides. To prevent contamination from squamous cells from the skin, gloves should be worn. Section thickness is also very important. For immunohistochemical staining, tissues should be cut at 3 to 4 microns in thickness. Sections should be picked up in the same area of the slide, usually in the center, to ensure proper coverage of the reagents. It is good practice to avoid placing the tissue on the slide edges, as the edges are commonly missed by reagents or it dries out. These areas may show false negative results or nonspecific staining. You'll see in these staining methods that they talk about air drying. And we'll talk about these staining methods at the end of the lecture, but there are steps that talk about air drying. And air drying the sections in front of a cool fan overnight is optimal, but it's not really a realistic expectation in the histology laboratory. Tissues can be placed into a 50 to 55 degrees Celsius oven for 30 minutes instead of the air drying. It's important that the oven never reaches above 60 degrees Celsius. So nuclear and cell surface markers are sensitive to high temperatures, and if they're destroyed, this will lead to a false negative or a weak staining. So 50 to 55 degrees Celsius in the oven for 30 minutes, never reaching above 60. So this is a photo from Dr. Carson's book showing a wavy section that resulted from poor tissue adherence. This is caused by improper microtomy technique. This is another photo showing 3,3-diaminobenzidine chromogen, so this is the DAB chromogen, that is trapped underneath the tissue. This is obviously an unacceptable sample for use. Epitope retrieval methods are used when hydrogen bonds formed during fixation need to be broken down. Overfixation of tissue by formaldehyde can result in an antibody not being able to access its epitope. And if this happens, that results in a false negative staining of the tissue sections. So here is a list of the advantages of epitope retrieval. So there is the ability to further dilute antibodies, exposure of epitope sites not previously detectable, more intense staining reactions with decreased incubation times, more uniform staining, decreased background staining, day-to-day consistency of stains, and the possibility of better standardization within the laboratory. There are two main categories of epitope retrieval methods: heat-induced epitope retrieval, or HIER, and enzyme-induced epitope retrieval, or EIER. We'll talk about each of these methods now. So the heat-induced epitope retrieval, or HIER, is a method of epitope retrieval that uses heat. It's not currently known actually exactly why the heating method works. High heat is the most important component of this method, but the composition of the retrieval solution is also critical. The original retrieval solutions, which were saturated lead thiocyanate and 1% zinc sulfate, had the potential to be toxic, so they've been phased out. Currently, the two most widely used retrieval solutions for the HIER method are sodium citrate buffer and EDTA. Other methods are distilled water, glycine, hydrochloric acid, and 3 molar urea solution. pH is very important in antigen retrieval. In antibodies and surgical pathology, high pH solution is recommended. Tris hydrochloric acid or sodium acetate buffer at a pH of 8 to 9 is suitable for most antigens. Nuclear antigens like estrogen receptor ER and retinoblastoma protein RB have optimal staining at a lower pH. There are other methods of heating that are available, and these include using a modified pressure cooker, a laboratory oven, a vegetable steamer, and circulating water baths. A standard method should be used in every laboratory, but at least 2 methods should be available for use. Enzyme-induced epitope retrieval, or EIER, uses a proteolytic enzyme to expose epitope sites. Since HIER was created, EIER is used less frequently. There's no universally used proteolytic agent, so a variety of solutions have been used. These include 0.1% pronase in 0.5 molar tris buffer, 0.6% physin, 0.1% protease in PBS, and 0.4% pepsin and 0.01 normal hydrochloric acid. With each of these proteolytic agents, digestion times may vary. Proteolytic enzyme digestion usually decreases nonspecific staining, and it may increase that nonspecific staining if not used properly. It can weaken specific staining, which can create false negative results. Overdigestion means when there is distorted tissue morphology, which results in poor staining. This is caused by too long of incubation times or if the enzyme concentration is too large. For effective EIER methodology, digestion time should be controlled and the same temperature used. Usually a temperature of 37 degrees Celsius is commonly used for the incubation of enzymes. Just using EIER alone can of course reduce the quality of tissue morphology. Because of this, it is useful to combine both methods of the retrieval of epitopes. HIER is performed first, then the tissue is rinsed, and then EIER for a short time, usually around 1 to 5 minutes. Okay, so let's talk about our methods of visualization for these techniques. These methods are immunofluorescence and enzyme immunohistochemistry. Immunofluorescence is actually the oldest of the immunohistochemical techniques. It is used to visualize antigens in tissue sections or in live cell suspensions. This technique uses something called a fluorochrome. This is a dye that absorbs light and then emits its own light at a longer wavelength, and this light emission is called fluorescence. When the fluorochrome dye is conjugated to an antibody, which means it's combined with it, so the fluorochrome dye combines with the antibody, this allows this labeled antibody to be easily seen. So the antigen reacts with that labeled antibody, and of course it can be seen. The most commonly used fluorochromes are fluorescein isothiocyanate, or FITC, and rhodamine. Both the rhodamine and the FITC absorb UV light and emit light that is visible to our eyes via a fluorescent microscope. All antigens can be detected with immunofluorescence. This method is very sensitive, specific, and very simple. In most histology laboratories, skin biopsies and frozen kidney specimens are examined with this technique. Tumor differentiation is performed usually with enzyme immunohistochemical techniques with light microscopy. We'll talk about enzyme immunohistochemistry on the next slide. For enzyme immunohistochemistry, the enzyme in presence of a substrate and a chromogen allows the visualization of the target antibody. Enzymes such as alkaline phosphatase, beta-galactosidase, glucose oxidase, and horseradish peroxidase are used as markers for this technique. Horseradish peroxidase, or HRP, and alkaline phosphatase, or AP, are most commonly chosen for this. HRP is most commonly used and is considered a workhorse because it consistently produces a large quantity of slides. HRP in the presence of hydrogen peroxide as a substrate and a chromogen such as DAB or AEC will identify sites of antibody binding by forming a compound that is colored. AEC is soluble in organic solvents requiring an aqueous mounting medium where DAB stained slides are mounted with synthetic resin. AP is demonstrated by the use of naphthol AS phosphate as a substrate and chromogens such as fast red violet LB, fast red TR, or fast blue BBN. AP is light and heat sensitive, and this can lead to inconsistency in staining. Semi-permanent chromogens that are compatible with AP allow the use of permanent mounting medium but do not require dehydration and air drying before cover slipping. If the specimen is not properly dehydrated, the AP chromogen can break down and precipitate across the tissue. Red chromogen signals can assist in identifying antigen sites in tissues containing melanin pigment, which may lead to obscuring a brown signal. If hematoxylin is used as a counterstain, Mayer hematoxylin is recommended, and this is because it does not contain alcohol. If other hematoxylins are used, such as Harris hematoxylin, which contains alcohol, with AEC or AP chromogens, the reaction product is dissolved and removed from the section because of that alcohol. This obviously leads to a false negative reaction. So this is melanoma of the eye tissue, stained with HMB45 using an alkaline phosphatase or AP red chromogen, which helps to contrast with the melanin pigment. There are a variety of techniques to detect the presence of antigen in tissue samples or the presence of antibody within the patient's serum, so in their blood. The methods we'll be discussing are direct, indirect, unlabeled or soluble enzyme immune complex, avidin biotin, and polymer and monomer. Typically, the words direct and indirect refer to methods used with immunofluorescence techniques. Unlabeled, soluble enzyme complex, and avidin biotin methods, or ABC, usually refer to visible light microscopy methods. So indirect methods, a labeled antibody with known specificity is used to identify antigens within a tissue sample. So the antibody is labeled with FITC or other fluorescent dyes, or it can be labeled with an enzyme such as HRP, so horseradish peroxidase, or AP, alkaline phosphatase, or glucose oxidase for a reaction with a chromogen for visualization under a light microscope. An example of this in the histopathology lab is the detection of immune complex deposition in kidney biopsies using direct immunohistochemical techniques. In the indirect methods, a patient's serum, so blood, is added to tissue sections that have known antigens. This tests the patient for the presence of antibodies to those specific antigens. An example of this is to test for anti-nuclear antibodies. Anti-nuclear antibodies are those that attack the nucleus of a cell. Presence of these indicate an autoimmune disorder. A labeled antibody must be used to detect bound antibodies from the patient's serum. Unlabeled or soluble enzyme complex method is a three-step method that uses a primary antibody, linking or secondary antibody, and soluble enzyme anti-enzyme complex. The primary antibody and the enzyme anti-enzyme complexes must be made from the same animal species in order for the second antibody to link them together. The most common techniques in these categories use either a PAP or AP anti-AP immune complex. Avidin has a super high affinity for the vitamin biotin. It has a way higher affinity for biotin than the affinity for antibodies for most antigens. The binding of avidin and biotin is irreversible, essentially. Avidin is isolated from egg whites and was first used in the ABC methods. So in the ABC method, which stands for avidin biotin enzyme complex, the primary antibody is followed by a biotinylated secondary antibody, which serves as the linking antibody. The next step in the ABC method is application of a preformed avidin biotin enzyme complex. The ABC method has a high sensitivity. The antibodies in this method may be used at higher dilutions than in other techniques. Dextran polymer and monomer techniques have become a standard for clinical immunohistochemical staining. Polymer enzyme molecules such as horseradish peroxidase, HRP, or alkaline phosphatase, AP, have been fused with secondary antibody. Monomer techniques use a single strand, which allows for more sensitivity and penetration at the antibody binding site. Like with every test or method in the laboratory, quality controls are necessary with immunohistochemical methods. A positive control, meaning a control that we know has a positive reaction with whatever method is being performed. So an optimal control is one that is prepared in the laboratory in the exact same conditions as the patient's tissue sample. The best practice is to use a multi-tissue control that contains both a positive control and negative control tissue for an antibody. Negative controls are run by substituting the primary antibody with either non-immune serum from the same species or with the diluent used for the primary antibody. If the diluent is used, the negative control won't detect nonspecific binding of animal serum, and any staining observed is the result of either endogenous peroxidase activity or the binding of other antibody reagents to the specimen. So when I say endogenous peroxidase activity, I mean the normal natural activity of peroxidase in the tissue. Okay, so let's talk about the evaluation and validation of antibodies we use in the histopathology laboratory. An antibody specification sheet is provided with any antibody that is purchased for use. It contains information about the antibody or the antiserum. The sheet usually includes information on the expiration date, the recommended storage conditions, both long and short-term storage temperatures, the concentration of protein and the suggested working dilution ranges, pre-treatment solutions, expected positive and negative tissue types, references to publications and applications for the antibody, and the antibody type, meaning if it's in vitro or for research only use. Antibodies can come into the laboratory in two different forms, pre-diluted or concentrated. Pre-diluted antibodies are ready-to-use solutions that can be purchased from a vendor. The vendor takes a concentrated antibody and dilutes it down into an optimal dilution for use. Because of this dilution, the shelf life is decreased, meaning its expiration date is going to be shorter. Pre-diluted antibodies must be validated to ensure they work properly before using it on a patient's sample. Concentrated antibodies are ones that are supplied in an undiluted form. The user has to dilute these concentrated antibodies in order to achieve an optimal dilution for use. The concentrated antibody is considered more economical because it has a longer shelf life than in the pre-diluted antibody form. Most concentrated antibodies can be snap frozen using liquid nitrogen and stored at a negative 70 degrees Celsius. That cannot happen for the pre-diluted antibodies. If the concentrated antibodies come in a solution form, it's best practice for the user to only dilute the smallest amount of antibody needed. This helps to keep costs down. Storage of antibodies is important to ensure the antibody works properly and also for economical reasons for the laboratory. Polypropylene, polycarbonate, and borosilicate glass containers are recommended for the storage for antibodies. And this is because these materials have low protein absorptive properties. So it's not going to absorb the protein within the solution. If the solution of antibodies has very low concentrations of protein, bovine serum albumin, so that's from a cow, that is 0.1 to 1% in concentration, can be added to reduce loss through polymerization and absorption of the storage container. In terms of storage temperatures and conditions, it's imperative that the recommendations of the antibody manufacturer be followed. Most immunoperoxidase methods have two blocking reactions. The first blocking reaction is from the use of 3% hydrogen peroxide. This is usually prepared in absolute methanol. This helps to block tissue endogenous peroxidase activity. And again, endogenous peroxidase is the naturally occurring peroxidase activity within the tissue sample. If the tissue sample has a lot of red blood cells, this first blocking reaction is absolutely necessary. The second blocking reaction is for nonspecific background staining. So we're trying to get rid of that nonspecific background staining. So this nonspecific background staining can occur as a result of antibody attachment to highly charged collagen and connective tissue elements within the sample. If the first protein solution applied to the tissue is the primary antibody, nonspecific binding can occur. The secondary antibody will bind to this nonspecifically bound primary antibody and will react with the substrate chromogen. This results in nonspecific positive staining. To prevent this nonspecific staining, an innocuous protein solution should be applied to the tissue before the primary antibody is applied. Secondary antibodies that are usually called multi-link or universal refer to antibody cocktails that are raised in different species. They are commonly biotinylated anti-mouse IgG and IgM and anti-rabbit IgG. Goat antibodies can also be used. It's important to know the species and the secondary antibody mixture to ensure that the targeted antibody is compatible. So these multi-link secondary antibody mixes are helpful because it prevents the problem of having to stock very specific link antibodies. You can use this because it's a mixture of different species. The reaction product from diaminobenzidine, or DAB or DAB, can be intensified by a few different methods. Heavy metals can be used for this by supplementing or following the incubation medium with reagents like 1% cobalt chloride. With this method, there is a risk of an increased level of background staining. Imidazole can also intensify the DAB reaction. Adding imidazole to the DAB incubating medium at a pH of 7.6 increases the sensitivity. It also inhibits pseudoperoxidase activity of hemoglobin, which is in red blood cells. Osmium tetroxide is another DAB intensifier, and the difference here is that it's used after the DAB reaction takes place, not before. The pH of the buffer used in immunohistochemical staining must be maintained and regularly checked. Any time that there is a new lot of buffer that is used or prepared, the pH must be checked and documented before use. Usually 2 buffers are used during the staining process for the immunohistochemical stains. These are PBS and tris-based solution, also called TBS. Both of these buffers can be used with an HRP detection system, so horseradish peroxidase. For alkaline phosphatase or AP detection systems, only TBS should be used. Adding a wetting agent like Tween 20, Triton X, and Bridge 35 helps with the even distribution of the reagents during staining. This addition also helps to slow down bacterial growth and improve the rinsing steps of the staining procedure. So we are going to start talking about how we use antibody stains, but first we need to talk about some relevant terms that are used within the chapter. So a neoplasm is a new growth of tissue. This is where the multiplication of the cell is just uncontrolled, and it can be either benign or malignant. And we'll discuss benign and malignant terms here in just a second. A tumor is a swelling and is one of the cardinal signs of inflammation and morbid enlargement. The term tumor is often used to mean neoplasm. A benign neoplasm means that there is a lesser degree of anaplasia than malignant neoplasms. Benign neoplasms are surrounded by a fibrous capsule and don't produce metastasis or any significant invasion of other tissues. Malignant neoplasms are those that invade nearby tissue and metastasize. Malignant neoplasms typically show degrees of anaplasia, which is a lack of cell differentiation. Some features of this are variation in size and shape of the cells, dark nuclei, giant cells, and a disturbed orientation. Differentiation is a term used for distinguishing one thing from another and the extent to which neoplastic cells resemble comparable normal cells. The primary site is the site at which a neoplasm first arises. Metastasis or metastasis is the transfer of disease from one organ or part of the body to another not directly connected with it. An example would be if someone has lung cancer and the cancer metastasizes to the brain. 2 separate tissue types not near each other, right? But the primary would be the lung and the lung tissue, and then it metastasized to the brain. Antibody stains are used frequently in pathology to identify a neoplasm and also to determine if the neoplasm is benign or malignant. Also, if the neoplasm is metastatic or if it is primary. The type of neoplasm is determined by the germ cell layer, so whether it's the endoderm, the mesoderm, or the ectoderm from where it is derived. So types of neoplasms include carcinomas, which start in the epithelial tissues of the body, sarcoma that develops in the connective tissue, lymphomas, which are in the lymphatic system, melanoma, which develops in the skin, mesotheliomas, which develop in the mesothelium, which is the tissue that lines the lungs and the abdomen, neuroendocrine tumors, gliomas, which are in the brain and also in the spinal cord, and germ cell tumors, which develop in the reproductive organs. There are three immunohistochemical techniques used for light microscopy that we will be discussing within this lecture. So these are the basic PAP immunoperoxidase procedure, ABC immunoperoxidase procedure, and the HRP enzyme-labeled polymer procedure. The basic PAP immunoperoxidase procedure is used for the localization of tissue antigens. So it's going to show us those tissue antigens. This method uses three reagents: a primary antibody, a secondary antibody, and a PAP complex. The PAP complex is composed of the enzyme peroxidase and an antibody against peroxidase. The primary antibody is specific for the desired antigen, so that means that it's specific for the antigen that we're trying to look for. The secondary antibody can bind to both the primary antibody and to the PAP complex. For this procedure, tissue sections should be cut 4 to 5 microns in thickness and mounted on positive charged poly-L lysine coated or siliconized slides. So these can be commercially purchased. They are to be air dried overnight, or you can dry them in the oven at 50 to 55 degrees Celsius for 30 minutes. Just make sure that if you choose the oven route that the temperature never exceeds 60 degrees Celsius. Quality control must use a tissue that is positive for the antigen in question. To perform the basic PAP immunoperoxidase procedure, you want to take the slides immediately from the oven and place them in 3 changes of xylene, and each of those changes you need at least 5 minutes each in them. Then from that xylene, you want to place the slides in absolute alcohol. For this step, you want to do 2 changes of that absolute alcohol for 3 minutes each. Then they need to be treated with 1% hydrogen peroxide and absolute alcohol for 30 minutes at room temperature. After that 30-minute period, you want to wash in 2 changes of 95% alcohol, then do a distilled water wash. Now, at this point, you want to use an epitope enhancement method of choice if indicated. Then rinse with distilled water, then rinse with working PBS solution, and then the following steps need to be performed in a moist chamber with the slides flat. You want to wipe around each section to remove any excess buffer, and then apply a blocking serum for 10 minutes. After this 10-minute period, you want to shake off excess blocking serum. After the excess blocking serum is shaken off from the slides, you want to flood the slide with primary antibody and incubate at room temperature for 1 hour. After the 1 hour period, you want to wash with working PBS solution and then flood the slides with it for 10 minutes. After the 10 minutes is over, you'll want to wipe the slides and apply working dilution of linking antibody. This step has a 30-minute incubation. After that 30-minute incubation, slides should be washed with PBS and then wiped, and then PAP should be applied. Once the PAP is applied, it needs a 30-minute incubation. And after this incubation step, it needs to be washed well with PBS, followed by a distilled water wash. Next, the sections need to be developed with AEC for 30 to 40 minutes, then washed with several changes of distilled water. Then it needs to be counterstained with Mayer hematoxylin for 15 minutes, and after that 15-minute counterstain, running water wash for 15 minutes. After this point, the slides can then be mounted with aqueous mounting medium or crystal mount. The ABC immunoperoxidase procedure, the ABC stands for avidin biotin complex immunoperoxidase procedure, is used for the localization of tissue antigens, so antigens within the tissue. This particular method utilizes 3 reagents: primary antibody, biotinylated secondary antibody, and ABC. And ABC, this is the enzyme peroxidase conjugated to a complex of avidin and biotin. The primary antibody used is specific for the desired antigen, so whatever antigen we're looking for. The secondary antibody is conjugated with biotin and can bind to the primary antibody and also the ABC. For this procedure, tissue should be fixed in 10% neutral buffered formalin, or zinc formalin can be used. They should be cut 3 to 4 microns in thickness and mounted onto positively charged slides. The tissue section should either be air dried overnight or oven dried at 50 to 55 degrees Celsius for 30 minutes. Just as a note, it should never exceed 60 degrees Celsius. Quality control or QC for this procedure should be a tissue that is positive for the antigen in question, and a negative control should also be run. So again, there are 3 reagents needed for this procedure: the primary antibody, the biotinylated secondary antibody, and the ABC complex. I discussed this already on the previous slide, so the information here doesn't need to be repeated. There are 2 parts to this procedure: the building of an ABC-HRP complex and the actual procedure. So the following steps are necessary to build the ABC-HRP complex. To make the peroxidase block, 3% hydrogen peroxide solution in DI water or methanol, and this is used to block tissue endogenous peroxidase. The protein block, so blocking serum, it's normally 10% solution that is used to block the normal serum from species in which the secondary antibody is raised. Primary antibody, so antigen that is targeting the desired epitope to be demonstrated. The biotinylated secondary antibody, so this is a complex that reacts with the primary antibody to build a bridge between it and the staining complex. This link is in commercial kits. An enzyme label, so this is a complex conjugated to an enzyme, and it contains HRP. It's called label in commercial kits. Chromogen, this is a chemical that reacts with that label so that the antibody complex can be visualized as a color reaction product. And then the counterstain, used to demonstrate tissue morphology without interfering with the color reaction of the chromogen. To perform the ABC immunoperoxidase procedure, you want to place the tissue sections in xylene, 3 changes at 5 minutes each of the xylene. Then place an absolute alcohol, and this is 2 changes of absolute alcohol for 3 minutes each. If the tissue is fixed in zinc or B5 solution, it should be treated with iodine and sodium thiosulfate, and the reason for this is it removes mercury pigment. After this treatment with iodine and sodium thiosulfate, it should be washed with tap water, then distilled water, and then with absolute alcohol. After this, it should be treated with 1% hydrogen peroxide and absolute methanol for 30 minutes. And this should be, the step should be performed at room temperature. After this step, wash with 2 changes of 95% alcohol and then rinse with distilled water. Then a rinse with working PBS solution. So after this, the following steps to be done need to be in a moist chamber with the slides flat. Then the blocking serum needs to be applied and allowed to stand on those slides for 10 minutes. And then the blocking serum should be the excess of it should be shaken off of those slides. After that's done, the primary antibody should be applied and allowed to incubate for 60 minutes, so an hour. The blocking serum should be left on the negative control slide. After this 60-minute or 1-hour period, the slides should be washed with buffer, then flood with buffer, and then leave them for 10 minutes. After that 10-minute period, the slides should be wiped, and then a diluted biotinylated antibody should be applied and then incubated for 30 minutes. After that 30-minute incubation, the section should be washed well with buffer. Then the slide should be wiped and apply Vectistain Elite ABC reagent, and then incubate for 30 minutes. After 30 minutes of that incubation with that Vectistain Elite ABC reagent, it should be washed well with buffer and then washed well with distilled water. After that distilled water rinse, the section should be developed with AEC for 30 to 40 minutes. And after this 30 to 40 minute development, it should be washed well with distilled water. Then Marihematoxylin should be used as a counterstain, and this is for 15 minutes. After that 15-minute period, the slide should be washed again with running water for another 15 minutes. Once that's done, you want to mount it with aqueous mounting medium or crystal mount. Following the ABC immunoperoxidase procedure, a positive reaction will be brick red in color. This photo shows plasma cells in the section of gastrointestinal tissue using the ABC method. The HRP enzyme-labeled polymer procedure is used for the localization of antigens within tissues. This method uses a primary antibody and HRP enzyme-labeled polymer secondary. The primary antibody in this method is specific for the target antigen, so the antigen that we're looking for. The polymer has an affinity for that antibody. Tissues for this procedure should be fixed in 10% neutral buffered formalin and cut at 3 to 4 microns in thickness. The section should be mounted on positively charged slides. These can be bought. Tissue sections should be allowed to air dry overnight. Another alternative would be to dry them in an oven at 50 to 55 degrees Celsius for 30 minutes. And after that 30-minute period, slides should be placed in front of a cool fan for 15 minutes. So that can be done, or they can just be air dried overnight. Quality control tissue should be 2 tissue sections, one that is positive and one that is negative. So again, 2 main reagents here, the primary antibody and the HRP enzyme-labeled polymer secondary. Tris buffered saline solution is also used along with the DAB chromogen and 3% hydrogen peroxide. To perform this procedure, the slide should be placed into xylene, so 3 changes of this at 5 minutes each with agitation. Then the slide should be placed in 100% alcohol, 3 changes of this at 5 minutes each with agitation. Then it should be placed in 95% alcohol, this time 2 changes of that 95% alcohol for 5 minutes each with agitation as well. After this step, slide should then be rinsed in distilled or DI water for 1 minute, and DI water is deionized water. After this rinse, the slide should be placed in 3% hydrogen peroxide for 10 minutes. If there's a specimen like bone marrow or a tissue that has a large amount of red blood cells present in it, you may need 20 minutes instead of that 10-minute period. After this step, slides should be rinsed again for 1 minute in distilled or DI water. The pressure cooker should be prepared according to the manufacturer's recommendations and follow their established protocol for performing antigen retrieval. Slides should be removed from the pressure cooker and let to sit for 20 minutes. Following that 20-minute period, there should be a 1-minute rinse in either distilled water or DI water, then a rinse in TBS solution with a pH of 7.6. That buffer should be, the excess buffer should be tapped off and make sure that the tissue is not allowed to dry. Then the primary antibody needs to be applied and then incubated in a moist chamber for 25 minutes. After that 25-minute period, it should be rinsed well with that TBS solution and then placed into a Copeland jar for 5 minutes. The excess buffer should be wiped off, then the secondary should be applied, and then the slide should then be placed in a moist chamber for 10 minutes. Then rinse well with TBS and then place in a Copeland jar for 5 minutes. After this 5-minute period, excess buffer should be wiped off, and then of course you want to apply the DAB chromogen solution and again place in a moist chamber for 5 minutes. Then it should be rinsed with distilled or DI water. Mayer hematoxylin uses the counter stain for 1 to 3 minutes, and then after that you want to cover slip. A positive reaction with the HRP enzyme-labeled polymer procedure is brown to dark brown in color. This photo on the right here shows prostate cancer stained with the P504S antibody. The chromogen in this is 3,3-diaminobenzidine. Alrighty, so that wraps it up for our immunohistochemistry lecture. If this video helped you out, go ahead and give it a like, and please make sure to subscribe to my channel for more educational laboratory content. Alright, until next time.